Hello again. Welcome to Lesson 10 for Honors Neuroscience. Today we'll be talking about receptor types. And this is going to be a very good lead-in into the pharmacology section, in which we're going to see how really interesting and complicated, and in a lot of ways, sort of wacky things can actually get when we're dealing with CNS function. But it's really interesting topics, and so I think you guys are really going to like this as we move forward. So I start off with this first slide because I think it illustrates quite nicely both the complexity and a couple of the major points that I want to get across here for neurotransmitter receptors. So you can see we have a nice neat little table, and on the left column we have the neurotransmitter, so these should look familiar to you by now. We've got acetylcholine here, norepinephrine, epinephrine, serotonin, glutamate, and GABA down here at the bottom. So these are all neurotransmitters that we talked about. We talked a little bit about their function. Now, we're not going to go through everything in the table. The main reason why I chose this table was for this column here. And here we have mechanism of action. Now, what you're going to notice is that for each neurotransmitter, it says something about what we call a mechanism of action. Now, a mechanism of action basically means the way in which some kind of effect is achieved. So how we achieve an effect. And in this particular instance, we're talking about the type of receptor activation. So this is why I showed this table. For example, here for acetylcholine, we can see that it says primarily direct through binding to chemically gated channels. All right, so we're going to be talking about exactly what that means, that we have a chemically gated channel, and the fact that this says primarily direct gives us a clue that it's not always direct, because there's actually a different type of receptor that acetylcholine can also bind to. So it can bind to two very different types of receptors that confer very different functions in cells. Well, here we have norepinephrine is indirect through what we call G proteins and second messengers, and that's something else we're going to talk about. And you can see similar sorts of other things here. Serotonin, for example, primarily indirect through G proteins and second messengers, and again, primarily because it's not exclusively. And in fact, there are other types of serotonin, one specific type of serotonin receptor that is a chemically gated channel. So the point here is that we have a lot of variety in terms of the type of receptors that these neurotransmitters are going to bind to. And that variety makes all the difference when it comes down to function. So what are the major receptor types? We're going to talk about two big ones. There actually is more than two, and it depends on how specific you want to get and how much biochemistry you want to get into and how much let's say enzymatic function you want to get into, but for us we're going to keep it simple and we're going to talk about two major types. The first type is called an ionotropic receptor. So this is what we think of as a ligand-gated ion channel. Now the key words here are ion channel, okay? Because the fact is that these receptors are very fast acting and they actually activation of these receptors allows ions to flow through the receptor into or out of the cell, as it turns out. And the second type is the metabotropic receptor. Now, for our purposes, we're going to be talking about GPCRs, so G protein coupled receptors, and we're going to talk about G proteins and what that means very shortly here. But metabotropic is named as such because they affect many metabolic processes. And as you're going to see, one of the reasons for that is because activation of these receptors can initiate a series of very complicated and complex events that ultimately can lead to the activation of many different types of enzymes and proteins. So, both types of receptors, whether we're talking about ionotropic or metabotropic, can cause excitatory or inhibitory sorts of actions. It just depends. It depends on several factors. And this is something that you're going to see firsthand. But before I get to my explanations, I really want you to take a look at this link here. This link actually will show you an animated version and a brief explanation of how each receptor type actually functions and how they're activated and what happens after they're activated. This is going to help you tremendously in listening to my explanation of the events, okay? So take the extra minute to click on the link. I think it's maybe like a 3 or 4 minute video, but I really want you to do that first, okay? When you're finished, go to the next part. Okay, so now that you've watched your video, I'm going to go through the basic big points of the difference between the ionotropic and GPCRs, or the metabotropic G-protein coupled receptors. And we're going to be talking about a couple of examples and to kind of get a better picture for how this happens, all right? So we're going to start with ionotropic, and here you can see that we have a receptor. This is embedded within a cell membrane, right? So this would be the extracellular region. This is intracellular, so this is in the cell. And here we have a bunch of ions floating around the outside, and we have a little green bubble. That is a neurotransmitter. Yes, you're correct if you're assuming that neurotransmitters don't really look like this, but for our purposes in drawing schematics and trying to make things very identifiable and easy, we often use these sorts of shapes, okay? So even though they don't really look like this, it's not too far off in terms of the channel structure, right? Now, in an ionotropic receptor, so in panel A here, this one on the left, a ligand binds, okay, in this case we're talking about neurotransmitters. Now I say ligand because we could also be talking about a drug, and we're going to talk about that later, all right? But in this case, a neurotransmitter is going to bind to the receptor, and it's going to cause a conformational change. Now this receptor is made up of a bunch of different protein subunits, and we know that proteins have three-dimensional structure, right? So what happens is when the neurotransmitter binds to this channel, this is actually going to cause a conformational change that's going to cause the channel to open, and ions are then going to be either permitted or restricted from flowing in and out of the pore, depending on what type of channel it is and what's binding to it, okay? So fast action, neurotransmitter binds, we get a change in the shape. The change in the shape will activate, for in this example here, the receptor, which causes a pore to open, and the neurotransmitter will flow from the extracellular space inside the cell, okay? Or the ion, excuse me. In contrast, metabotropic receptors function very differently. Now, there are a couple of things in common, okay? G-protein coupled receptors, so again, that's the type of metabotropic receptor we're going to focus on here. Now, there's tyrosine kinase receptors and a couple other things that sort of are separate categories, so we're just going to think of metabotropic receptors as GPCRs, okay, and that's the ones that that's the example that we're going to use here. Very similar mechanisms of action with the others, anyway. So here we have a neurotransmitter that's going to bind to this receptor. It's also going to cause a conformational change, okay. So similarities, we have protein that spans the membrane here in between the extracellular and intracellular. Neurotransmitter binds to it, it's going to activate it. Now the difference here is that we have what we call this G protein that's actually bound to the cytosolic or the intracellular portion of the receptor. Okay? Now this G protein is composed of 3 different subunits. It has an alpha, a beta, and a gamma. And what happens is in response to the neurotransmitter binding to the receptor, there is a conformational change and it causes the activation of the alpha subunit. So what essentially happens here is that a GDP, which is attached to the alpha, is exchanged for a GTP molecule. That GTP, once it's added to this alpha subunit, activates it. That alpha subunit activation is then going to trigger different types of effects in the cell. Alright? So we're not talking about activating a receptor and opening a channel per se. We're talking about activating a protein that's stuck to the inside of the receptor and the inside of the cell, and activation of that protein is going to cause part of it to jump off and activate other things later. Okay, so it's sort of like the baton game. So we're going to pass the baton here, and then there's going to be some other things that are going to happen here. Now this particular example actually shows an ion channel being activated. That's one thing that can happen, okay, in response to GPCR activation. We're going to talk about a couple different major mechanisms. But before we do that, let's talk about the structure, because these guys are very cool, all right? Very, very cool looking things when we actually get way down to the molecular level and examine how these receptors are put together. So there's a few words I want you guys to be aware of. You probably know a couple of these words from previous biology courses and things like that. But the structure can be described in a couple of different ways, right? Now we say it's tetrameric or it's pentameric. So when we're talking about an ionotropic receptor, a tetramer means it's composed of 4 parts, right? So tetra meaning 4. A pentamer is composed of 5 parts, so penta meaning 5. All right. So if you look on the right side here, this is a side view of what the receptor might look like, and there's a couple subunits back here that we can't see. Now this is looking at the same thing from the top, okay? And what we can see is that there's 1, 2, 3, 4, 5 different subunits. So this particular receptor is a pentameric receptor. Now each one is composed of a subunit, okay? So each part of this pentameric structure is one subunit. So it's a protein, okay, that has a helical structure that allows it to go through the membrane. Now each one of these subunits are connected to each other. So what we end up with is something that looks kind of like a barrel, right? That's actually embedded within the cell membrane. Pretty neat, right? Well, when the barrel is inactive, there are actually peptides and parts of proteins that block the channel, all right? So ions cannot pass in or out of it. Now this one's open. All right, so this is what it would essentially look like if it was activated. But under normal circumstances, it's closed, okay? So here we have a closed channel. Even though there's a bunch of ions floating around on the outside, they're not going to be able to get in, okay? Now, in this case, we have acetylcholine, all right? So acetylcholine is binding to what we call a nicotinic acetylcholine receptor. Now, the nicotinic acetylcholine receptor is one of the major, I call it a prototypic example of an ionotropic receptor because it's probably the most well-known, it's the most studied, and this is actually how we learned a lot about what we know about how ionotropic receptors work is through studying acetylcholine receptors, okay? Now, having said that, just because acetylcholine is binding to this channel, this ion channel, that doesn't mean that acetylcholine always binds to ion channels. And in fact, there's another set of receptors for acetylcholine that are called muscarinic receptors, and those are actually GPCRs. Okay, so acetylcholine can act on receptors that work as ion channels, such as this nicotinic one shown here, or they can act on GPCRs, right? So that's just something to be aware of. You're going to see that later. But in this case, okay, we have what we call a heteromeric receptor. And it's a heteromeric because it's composed of different types of subunits. And these different types are really just based on what they're made of, okay? It's based on their structure. We're not going to get into that, but it's good to know it. You may see this sometime. So, for example, we got a beta subunit here. Where's my pointer? A beta, we've got an alpha, we've got a gamma, etc. Okay, so a lot of times you'll see something that'll look like alphas and betas. Okay, so this is an alpha 4 beta 2 acetylcholine receptor. It's heteromeric because it's composed of alphas and betas. The fact that this is alpha 4 is just because of the sequence of this particular protein that makes up the subunit. So alpha 3 looks different, alpha 2 looks different than that one, okay? This is a homomeric receptor. This is an alpha 7, okay? Because these are all composed of exactly the same type of subunit. There's just 5 of them that are all linked together. Okay, so we've got heteromeric or homomeric. It's not terribly important for what we're going to be talking about in this class, but it's just something cool about the way that these receptors assemble that I wanted you to be aware of. Okay, but anyway, so we do have these structures. So these ionotropic receptors are composed of either 4 or 5 subunits that are all linked together. They form a barrel in the cell membrane. Okay, now, what are the different types of ionotropic receptors in terms of like their broad mechanisms of action? Okay, so I'm going to break this into 2 categories, and I'm going to tell you that things are never this simple. All right, you should know by now there's always going to be exceptions to things. But in general, okay, when we talk about a depolarizing ionotropic receptor, we're talking about a depolarizing ligand-gated ion channel. So a neurotransmitter is going to bind to it, and the channel is going to open, and it's going to cause the cell to get closer to depolarizing. Now, if you remember the action potential, what does depolarization mean? Well, it means that the neuron is going to fire an action potential. In order for that to happen, the membrane potential, okay, so the current threshold has to get more positive, right? Remember, the resting membrane potential for a neuron is about negative 70 millivolts, okay? And we need to get to about, I want to say negative 30 or so, and it depends what you read and what types of cells you're talking about and things like that. But we need to get more positive to initiate an action potential, right? Just to review, the action potential is when the cell fires. It gets an electrical current down the axon into the terminal and releases neurotransmitter, right? Okay, good. So depolarization means that the channel opens, right? Depolarizing means that opening of the channel is gonna push it more towards a positive membrane potential, okay? So the types of receptors that do this, all right, are glutamate receptors. The glutamate is the major excitatory neurotransmitter of the brain. So you remember we talked about that before. Now there's 3 different types of ionotropic glutamate receptors. So in other words, glutamate receptors that are ion channels. We have NMDA, AMPA, and kainate receptors. Now the only reason why they're named this is because these are different types of molecules that were found to be able to bind to this receptor, okay? When people were first studying them and were first trying to identify the receptor. And so they're named as such, right? Now, the NMDA and the AMPA are the 2 major glutamate receptors that regulate most of the excitability in neurons throughout the central nervous system, okay? It's usually NMDA or AMPA. Kainate plays a small role, and we're increasingly learning about kainate, but the precise roles of kainate are not as well known as those of the NMDA and AMPA receptor, okay? But the key point here really is that these are permeable to cations. So we're talking about things like sodium, okay, potassium, calcium. And this makes sense because if you think about it, one of the major ways that a cell can fire is by taking in sodium. Remember, the cell membrane potential is negative. For the cell to get more positive, we have to take in positive charges, right? And sodium is a positive ion. So the sodium is really important. Calcium also can help us to excite the cell as well. That's, again, another positive ion, okay? But not all are created equal. And we're going to see an example in a minute of why the NMDA receptor is a little bit different than AMPA, okay? Even though both are permeable to cations, right, positively charged ions, they're not equally permeable to all types of positively charged ions, okay? And this is really important. But by selectively permeable, what we mean is that they're only going to allow certain types of ions and not others, right? So for example, these glutamate receptors here are not going to allow chloride ions to enter, okay? They are selectively permeable, so they're only going to allow certain types, all right? Nicotinic acetylcholine receptors are also depolarizing. All right, so that means that they are going to allow sodium, and some will allow calcium, to enter into the cell and make the cell more positive, okay? So they're going to increase the probability that the cell is going to fire an action potential, okay? And we just saw the nicotinic ones. We talked a little bit about this already, okay? So for example, alpha-4 beta-2, you might see alpha-4 beta-2 nicotinic receptor. That just means that this is actually its structural composition, okay? But we're not going to memorize and learn all the structures or anything like that, so you don't have to worry too much about that. And we already talked about how important they are at the neuromuscular junction. And so the hyperpolarizing ones mean that they're actually going to make the cell more negative, right? So remember, we have to get positive to fire an action potential. So if we start letting in negative ions, what's going to happen to the cell? Yes, you're correct. Okay, it's going to get more negative. It's going to be less likely to fire. Okay, remember, this all goes back to the action potential. So GABA and glycine are the big ones, okay? In the central nervous system, it's really GABA. Glycine is more active in the periphery. And the reason, of course, that these are hyperpolarizing, right? Unlike the glutamate receptors that allow the passage of things like sodium and calcium, the GABA receptors, okay, the major inhibitory neurotransmitter of the brain, is selectively permeable to chloride ions, okay? So it's when this channel opens up, it's going to allow chloride ions to pass in, okay? And what's neat about these is that even though there's endogenous neurotransmitters that bind to them and allow certain ions to pass in and out, we can also alter the function of these receptors by giving exogenous stuff. So that's to say things that are made outside the body that we can take into the body. And what's one great example of things that are made outside of the body that we can take into the body that can affect receptor function? Are drugs. Okay, so I'm going to go through one example for each of these. One example of glutamate receptor. Okay, so this is an excitatory receptor and one important function that it's involved in, and then we're going to talk a little bit about GABA and drugs. Okay, and this is kind of just to put a face with a name, to kind of put a function with some structure and very detailed information that we're learning here. So when we talk about learning, and when we talk about learning and memory, and in fact, as you watch these videos and you study and you start to learn the material and you start committing it to your long-term memory, this is what's actually happening in your brain. All right. Now what you see here is two different receptors. Okay. We have an NMDA receptor and an AMPA receptor. Now the R at the end just means it's a receptor. Okay. Now remember, these are two different types of glutamate receptors. Now remember, we said these are selectively permeable to cations, but they're not all created equal, right? What's cool about this is that the AMPA receptor is very permeable to sodium, not really so much to calcium, okay? Now the NMDA receptor is permeable to both sodium and calcium, so it'll allow both of these to pass in. But there's an added wrinkle, okay? So under normal circumstances in the brain, okay, a cell might be hyperpolarized, and we need to depolarize the cell for it to fire an action potential, okay? So we need to make it more positive. Now, the NMDA receptor, even though it also helps to do this, can't really activate until the cell is more depolarized, until the membrane potential becomes higher, right? So make it more positive because it's blocked by magnesium ions. So the structure of this channel and the way that it's set up is such that magnesium will get in the way and will block it unless the cell has become more depolarized. So what happens with this cell is that when glutamate binds to the AMPA receptor, it causes the channel to open and it lets in sodium. Now, as sodium continues to pass into the cell, these ions make the cell more positive. And once the cell reaches a threshold positive charge, this NMDA receptor, in the presence of glutamate, remember glutamate also has to be bound, will then open because the magnesium will become dislodged from blocking the receptor, and it will then open and it will allow calcium to enter into the cell. Okay? So NMDA will let calcium in, but it only does it when the membrane potential is a little bit more positive, okay? And this is really important, okay? And this is important in long-term memory formation because one of the things that we know is that calcium in the body is necessary for consolidating, what we call consolidating memory, from taking things, any behavior or any sort of information that we learn and committing it into our long-term memory. Calcium is so important for so many different things. It allows the activation of different enzymes, okay? So it allows the release of neurotransmitters from the terminal. That's something we talked about earlier. So really, it's essentially impossible to overstate the importance of calcium in cells. By the way, calcium can also kill cells if you have too much of it being released. So there's delicate balance here, okay? But the major point here, and why I wanted to show this example, is because we have a situation where you have to have glutamate present, okay, but you also have to have a bit of a depolarization in order for the calcium to come in, okay, because this magnesium's stuck there and you got to bounce it out of the way. So this is for the NMDA receptor. This is triggered by activation of the AMPA receptor. So in order for appropriate learning to happen, you have to have a combination of very intricate receptor activation between these two different types. Pretty neat, right? They also have a word for this. It's called amplification. And so when you're learning something, what they notice is that you have all these AMPA receptors start popping up. Your cell starts making lots of AMPA receptors, and it starts shuttling them out to the cell membrane. And this is probably because we need more AMPA receptors to lower to make the membrane potential more positive, so that we can increase the function of our NMDA receptors, because those are the ones that are helping you memorize stuff. All right. Now, what about in the case of drug binding? Now we're going to switch gears and we're going to talk about a GABA receptor. Okay. And this is a particular type of GABA receptor. It's called a GABA A receptor. Okay? And what's cool about the GABA A receptor, remember we talked about GABA receptors as being selectively permeable to chloride ions. So that means they're going to inhibit, okay, the firing of the cell. Inhibit the firing of the cell. Well, there's lots of different drugs that have been found that actually have binding sites on the GABA A receptor. And they bind in a way that we call allosteric. Okay? This is something we're going to talk more about when we do get more into the pharmacology, but I wanted to introduce it here because it ties in really well with talking about the way that these receptors work. So here you can see a binding site for GABA. So the neurotransmitter GABA is going to come and it's going to bind in this particular place, okay? So it's going to form bonds, okay, with these molecules that are making up these proteins, these receptors. But we also have all these other places where different things can bind, okay? So here we have an ethanol binding site. We have a binding site for barbiturates and benzodiazepines. Those are two different types of drugs, and we'll talk a little bit about what those drugs do later, okay? But essentially, we can think of these as having sort of depressant and sedative effects. Sort of makes sense, because if we're going to inhibit the firing of neurons, we're probably not going to be doing things as quickly as we were before, right? So that's one of the reasons why sometimes you think of depressants, right, as kind of make you tired, you start moving slower, maybe you speak slower, right? And this all ties in with that as well, okay? But the reason why these work is because GABA is going to let in chloride ions anyway. But when you have some kind of allosteric modulator, like, for example, a barbiturate or ethanol, what they're going to do is they're going to bind to the receptor, and they're going to alter the function of the receptor either by increasing the number of times that it opens, okay? So allow it to keep opening a little bit quicker, because you have to remember, it's not like a neurotransmitter binds a receptor, opens it, and it stays open forever, right? It's going to close, and then the neurotransmitter is going to jump off. Then another one might come on, activate the receptor, it might open, let some ions on, then it might close again, the receptor and the neurotransmitter jumps off. So this jumping on and off between things that are binding and being released is always happening, okay? But what these guys do is they actually make this thing more efficient, and they make it either open more times or open for a longer period of time, okay? And when this happens, we're actually affecting the state of the cell. So the bottom line here, and really the important thing to kind of keep into consideration as we move forward in this course, is that it's not only the structure of the receptor. Of course that's important, right? Because now glutamate, for example, isn't going to have these same binding sites for these drugs. And that's the reason why these drugs have a very specific type of action in the body, is because of the type of receptor that they bind to and what that receptor does in normal physiology, and then how we actually are altering its effects, okay? But it's not only the structure of the receptor, it's actually where it's located. So, what do I mean, where it's located? Well, we talked about the rewarding effects of drugs and why drugs are rewarding. Well, how does this tie into receptors? And you can think of something like nicotine. So maybe you know somebody who smokes, and we say they're addicted to smoking, right? What does that mean? It means that they can't stop. Well, why can't they stop? One of the things we've learned so far is that there's a very potent reward and reinforcement mechanism in the brain. And that is regulated, of course, by this mesolimbic dopamine system. And here, we can have this ventral tegmental area, okay, of the midbrain. Remember, it's in the tegmentum, that has dopaminergic neurons that project to this region called the nucleus accumbens, okay, of the basal ganglia. And we know that if we activate this circuit, okay, in response to something, it's going to produce this dopamine release in this particular area that's going to reinforce behavior. It's going to make us want to do it more and more, okay? Now, it turns out that lots of receptors on these dopaminergic neurons in the ventral tegmental area, in the VTA, have nicotinic acetylcholine receptors on them, okay? Now, you may have guessed it. The nicotinic acetylcholine receptor was named as such due to its affinity for binding nicotine, okay? So affinity means how much it likes it. So nicotine binds very strongly and very well to the nicotinic acetylcholine receptor. So the bottom line is that these dopaminergic neurons that arise in the VTA, and project to the nucleus accumbens have lots of nicotinic receptors on them. So when you smoke a cigarette and you get this nicotine coursing through your body, it's a small molecule, it penetrates into the CNS, and it binds to these nicotinic receptors. Now remember that the nicotinic acetylcholine receptor is an ionotropic receptor. It's going to allow sodium to rush in. It's going to cause depolarization. So that increases the firing rate of these neurons, right? So that means we're releasing more dopamine in this area, and that's feeding our addiction, okay? So it's helpful, I think, at points to tie all these things together. And the reason why we're going through some of these small details is because when we get to talking about psychopathology in different parts of the brain, all these things are going to kind of start making sense and coming together for you, all right? So that's ionotropic receptors. Now we're going to talk about metabotropic receptors a little bit. These guys are also super neat. If you take a look on the left here, we have this sort of looks like a brain. It's not. This is actually a cell membrane. So here we have a bunch of phospholipid. There's, of course, there's going to be some cholesterol and other things in there. But here we actually have what we call a metabotropic receptor. And this particular one is a GPCR. Okay? So there's certain features of this receptor that I kind of want to explain so that we know what we're dealing with. These receptors are single polypeptides. They're very long, okay? Probably 450 amino acids, something like that, in that neighborhood. And they have 7 different transmembrane domains. Now, of course, transmembrane means it goes across the membrane, right? So transmembrane domains means they go back and forth, back and forth. And you can see it on the right side here, right? You can really see how these domains go back and forth. They have 1, 2, 3, 4, 5, 6, 7 of them here. Intracellularly, we talked about this already, we have our G protein. That's composed of these 3 subunits. It's the alpha, the beta, and the gamma, and it's bound by GDP here. So this GDP signals to us that this is in the inactive conformation, okay? Because remember, the way that we activate these guys is actually through GTP. So GTP takes the place of the GDP here. They have an extracellular N-terminus, okay? N-terminus is short for amino. Remember, that's NH2. Which really, well, I think you'll see it in the next slide, but sometimes it's NH3+. That's just because it's ionized at normal physiological pH. But NH2 is an amino group, right? Whereas COOH is a carboxyl group. So the C terminus means that we have a carboxyl at the end. And again, right here, okay, normally the amino group is an NH2. The carboxyl group is a COOH. The only reason why these are showing NH3+ and COO- is because at normal pH in the body, these are in their ionized form. Okay, but it's the same thing. So we have an amino side on the outside, we have a carboxyl side on the inside, and then we have the G protein bound to it. Okay, now this is coupled. So what that means is that the interior portion of this receptor has the G protein bound to it. Okay, now activation happens in a 3-step process. The first step is that we have a neurotransmitter that binds to some portion. And depending on which GPR it is, it's going to be a different binding site. Okay, just like the ionotropic receptors have different binding sites for different things, well, it's the same sort of deal here. Okay, but once this binds, again, we're going to have a conformational change. We're going to have activation of the G protein. Okay, and then once this GDP is exchanged for a GTP, this whole part here is going to dissociate into an activated alpha and a beta-gamma complex. Okay, so we say this is trimeric to begin with because it's composed of 3 parts. It then dissociates into the alpha and a dimer of the beta-gamma. Okay, so these two kind of stay together and move off, right? Now, what happens is 2 different possible things, okay? We can either have a quick activation or we can have a more complex activation, all right? So, G-protein gated ion channels represent the least complicated mechanism of G-protein activation, and this basically is when activation of a G-protein will cause the direct activation or inactivation of an ion channel that's next to it. Okay, so you can picture a cell membrane like this where we have a GPCR, okay, a receptor here that's a G protein coupled receptor, and we have another ion channel next to it. Okay, this happens to be a potassium channel, but it could be anything, right? There's all kinds of channels and receptors all throughout the membrane of a cell. So here, okay, this fast mechanism is direct action, and it's pretty simple because what happens is once the neurotransmitter binds to it, in this case we're talking about acetylcholine. In this case we're talking about cardiac myocytes, okay? So cardiac meaning heart, a myocyte is a muscle cell. So cardiac myocytes are regulated through acetylcholine function, okay? This is a muscarinic acetylcholine receptor. So unlike the ion pores, the nicotinic ones, okay, this is actually a G protein coupled acetylcholine receptor. Sorry. So acetylcholine binds, conformational change. We have GTP exchanged here, and that is going to trigger this alpha and the beta gamma to jump off. Okay. Now in this case, the beta gamma subunit is actually going to shoot over here, and it's going to interact with this channel on the intracellular side. It's going to cause the channel to shift its conformation and the pore to open, okay? And that's going to allow potassium to flow out, all right? Now this actually is a mechanism of slowing heart rate because what it really does is it keeps the cells from firing because potassium is actually going to flow out of the cell in this case, okay? So this is a potassium channel that's used to try to re-hyperpolarize the cell after it fires. Remember, the cell depolarizes, becomes more positive. Once it's full of positive charges in here, what happens? The potassium channel is open to let out some potassium ions to make the cell more negative again. So that's actually what's happening here. We're actually making the cell more negative. Not crucially important that you get that part for this, but it's just, this is the reason why, because you might have thought, well, why isn't it exciting it? It's not, because this actually allows potassium to leave the cell, okay? But you get the idea. So the alpha is activated, these dissociate, the alpha goes on to do other things, the beta gamma is actually going to come over here and it's going to activate this channel. Now these can also inactivate channels, all right? It just depends on which type of channel you're talking about, all right? So here, that's the money maker. Now, second messenger cascades are more complicated. So GPCRs, in addition to directly influencing an adjacent channel, they can also activate a whole host of other molecules, okay? And this is sort of like tag you're it, right? So what's going to happen here is we're going to have this ligand, again, neurotransmitter here is going to bind, conformational chains, GTP binds, right, exchanges the GDP out for GTP, and we have these dissociating off. So now we have our activated G-alpha subunit, the beta gamma go off to do their own thing. But now here what you see is that this G-alpha is going to trigger a whole series of events. And this is what we call the activation of a second messenger pathway. And these funny looking things here all basically are a series of different enzymes and activating molecules that lead to a particular cellular response. Okay? So here, this is actually adenylate cyclase. Okay? This is an enzyme that's activated by G-alpha. Okay? Which then catalyzes the conversion of ATP to cyclic AMP, okay, which is also a messenger that then activates protein kinase A. Now, protein kinase A is also an enzyme, okay, that's involved in some things that we'll talk about in a minute, okay? But the major point here, you don't have to, of course, memorize all of these things right now, but the important point to take from this is that we have activation of the G protein that then activates a whole different series of events, okay? So we have one enzyme activates another molecule, activates another one. This is what we call a second messenger pathway, okay? And it's called second messenger because from the point where we activate the first enzyme all the way to the end, everything in between are called second messengers, okay? So in this case, this cyclic AMP is actually going to be a second messenger molecule because it's involved in helping us go from the beginning to the end, all right? It's an intermediate step, all right? So this is the cool part about why these receptors are so interesting and why they can regulate so many different functions. This is just another schematic. This is a little bit simpler, so I can kind of take you through from beginning to end why this is important. So we have, again, GTP here. We have ATP being converted to cyclic AMP, making protein kinase A. Okay. Now, when we have protein kinase A activation, we have a whole bunch of different responses. And again, this is sort of the same thing that I talked about before. Now, the whole process, everything from beginning to end, is what we call a cascade. That's a second messenger cascade. The actual second messenger molecules are the ones that are involved in taking the first thing and turning it into the thing at the end. Okay, so we can have lots of different messengers, or we can only have one messenger. It just depends. There are tons and tons of pathways. Okay, I mean, you could spend an entire semester trying to memorize all the different pathways. So we're not going to do that, but we are going to highlight a couple as we go along that are interesting, that actually are targeted by certain drugs and things like that. All right, so again, here we have GTP activation. This is adenylyl cyclase. This is, again, I said this is an enzyme. Okay, in this case, it's bound to the membrane. And that's going to catalyze cyclic AMP production and protein kinase A. Now, you should have learned already about phosphorylation, but if you did not learn about phosphorylation or you don't remember phosphorylation, I'll give you the quick sort of important spiel about phosphorylation. It is critically important in the function of everything. Okay? Phosphorylation means that we have an enzyme. So in this case, it's protein kinase A, but it can be protein kinase C. Okay, a protein kinase is an enzyme that phosphorylates proteins. Phosphorylation means that we add a phosphate onto the protein. Okay, now when we add phosphates onto proteins, or we phosphorylate proteins, we change their biological function. Okay, and we can activate them by doing this, or we can inactivate them by doing this, just depending. Okay, so for example, if we have a second messenger cascade that ultimately leads to the activation of protein kinase A, now we have this pKa enzyme that can go and add phosphate groups onto lots of different stuff. Now, we might have a channel over here, okay, an ion channel. And if we phosphorylate that ion channel on the inside, it may cause it to open and allow passage of ions. It may cause it to become inactivated so that it can't function anymore. Okay? Phosphorylation of proteins can increase their function. It can change their function. It can allow them to be able to bind certain molecules that they couldn't bind to before. Okay? So just this one mechanism of phosphorylation unlocks an entire countless amount of different types of cellular regulation that we can undergo. All right, so that's why these pathways are super important. So this is just one more example. I want to talk just quickly about the different types of G protein cascades that there are, because there's several different ones. Again, sort of emphasizing also that it's very slow. All right, so when we talked about the ionotropic receptors, it's something that happens very quickly, right? It's the fastest action. The ionotropic receptor opens, the ions flow through. It's very direct, it's very quick, okay? When we have the GPCR action, where the G protein, directly activates or inactivates the channel, that's pretty quick too. It's not quite as quick as having an ionotropic receptor, okay? But it's a little bit slower, okay? But still not too bad. Now, this mechanism of activating this whole series of enzymatic reactions, right, is going to be much slower. It's not going to happen very quickly. But the cool thing about it is that we can actually amplify our effects, all right? And we can regulate lots of stuff. And I sort of talked about this before. The one I didn't mention, and the reason why I put this slide here, is gene expression. So we can regulate the expression of different genes by activating these different pathways. Okay, so we have what we call the GI and the GS pathways. These are two different types of G protein pathways. And the GS is the one that I just showed you before, which actually leads to the activation of adenylyl cyclase, cyclic AMP, and protein kinase A. This protein kinase can phosphorylate lots of stuff. Okay, so this, in a way, you can think of as being sort of like an activating pathway, because it's going to activate PKA. It's going to cause lots of things to be able to be phosphorylated. Whereas with the GI pathway, what this actually does is it shuts off adenylyl cyclase. Okay, so this black bar here is actually showing that we're blocking this. So what we're really doing here is we're reducing the ability of ATP to get converted into cyclic AMP, which is required for PKA activation. So here, if we have GI protein receptor, okay, a GI PCR or GPCR, what we're going to be doing when something binds to this is we're going to be blocking this effect, okay? So we're reducing the potential to be able to phosphorylate stuff, all right? So phosphorylation is a key result of activation of this G sub S, and if we have the G sub I GPCR, we're going to block that effect, okay? We also have the GQ pathway, which, among other things, actually leads to increased intracellular calcium. All right, and we talked about how important it is to elevate intracellular calcium. All right, so I'm not going to get into all the details of what these guys are. It doesn't really matter. Okay, but just I want you to know of two major effects that we can get from these GPCRs. One of them is phosphorylation, which allows us to activate enzymes. It allows us to change the expression of different genes, alter the function of channels, and we have intracellular calcium increase, which can allow us to do many of those things as well. Okay, so there's a lot of overlap between these different systems. So you might say to yourself, if these second messenger pathways are so slow, why do we use them, right? Well, the reason why they're so great and they're so effective is because of amplification. What you can see from this pyramid diagram here is we have a transmitter that's binding to a receptor, all right? Now this is going to activate G proteins, which are then going to cause the G alpha subunits, okay, to go on and have their effects. So here we're using the same GS pathway. So we have adenylyl cyclase here being activated, right? So that's causing cyclic AMP to be produced, right? And then that's causing lots of PKA activation and lots of phosphorylation of a whole bunch of different potassium channels, all right? So the idea here is through the binding of just one receptor, we can cause lots of different effect, okay, because it's amplified every single step along the way, okay? So one binding on the receptor causing lots of response in the cell. So even though it happens slower, it's a good way to kind of amp up a type of particular response that we want a cell to have, all right? If that makes sense. All right, and so I'm going to leave you with this one. These are the different receptor families, just a few of them. They're not all of them. There's tons of them, and there's more receptors than it actually shows here, okay? And you can make a living out of trying to figure these all out. But the main reason why I put this on here was to talk about the concepts of divergence and convergence and how that really leads to integrated signaling. And we really already did this, okay? But just to kind of bring it full circle, we have acetylcholine here. Now these are G protein coupled receptors. So what you can see is that these muscarinic ones, unlike the nicotinic pores that let in ions, these muscarinic receptors are GPCRs. And there's a whole bunch of different types of them. And some might be GS, some might be GQ, some might be GI. So acetylcholine by itself, okay, can regulate cells in lots and lots of different ways, because it can not only open channels directly, but it can also activate different types of second messenger cascades, okay? And depending on which type of cell in which region, okay, that particular type of receptor is, it's going to cause a different effect, okay? Glutamate, same thing. Even though we talked about the glutamate receptors, the ionotropic ones, right, which are the NMDA receptor, the AMPA receptor, etc., there's also metabotropic glutamate receptors, okay? So glutamate can also activate GPCRs in addition to the ionophores, okay? So the divergent aspect of this, the fact that one neurotransmitter can activate a lot of different types of receptors, okay, means that we can have a host of different actions, okay, by a single neurotransmitter. And when you add these all up together, okay, what we see is that a lot of times multiple neurotransmitters will converge to affect the same system. So we already saw an example of how we can increase the firing of a cell, right? And we can also increase intracellular calcium, okay, which can cause us to phosphorylate channels, which will then increase the amount of ions that can come into the cell. So depending on what types of receptors are in which places, okay, we can have a bunch of different neurotransmitters that either converge on the same function or actually kind of sometimes can compete with each other or produce very separate actions. So the take-home message is it's really complicated, and we can achieve basically anything in lots of different ways by having a bunch of different types of receptors that are activated through different mechanisms with a bunch of different neurotransmitters. Okay, and what I gave you was a small overview of some of the different types of neurotransmitters and receptors. Right, and like I said, as we go forward, we're going to be highlighting certain ones that are specific to the exact topic that we're talking about, rather than trying to go through all of them, because frankly, there's just too many. And trust me, you don't want to do that. All right, so here's a quiz for you. I hope you enjoyed it. I hope you had as much fun as I did. I know this one was a little bit longer, but I'm sure that it was worth it. So have fun, and I will see you next time.